welcome to Flora and Friends, your botanical cup of tea, a podcast for plant lovers of any kind. We welcome guests to our botanical tea break to explore the history, science and meaning of plants for our lives. My name is Judith Lundberg-Felten. I'm a plant scientist, university researcher and founder of Flora-L Design, and I'm the hostess of your botanical cup of tea. Hello, friends of the Flora and Friends podcast. I'm delighted to be here with you again for our first episode of the year 2022. And my wonderful guest today is Uppsala-based artist Amanda Selinder. And I discovered Amanda a few weeks back when I visited Uppsala Art Museum at the castle. And she was one of the 6 stipend holders through the year that had worked with a project for Uppsala. And when I looked at the arts exposition, I was directly drawn to a big bunch of Petri dishes with fungal cultures that were hanging all over the wall, from cultures that were more black towards some that were more brownish, red brownish, beige. So, and beside of that, there was silk dyed with pigments from these fungi. And I found it so interesting that Amanda comes from her background from fine arts and uses biology to visualize biological processes. And I come with my background of biology and try to identify artistic ways in order to show what is inside plants through microscopy designs. So I wanted to talk to Amanda and see where her motivation came from, how she discovered the possibility to use with natural pigments, and she really unraveled to me the process that it took her to get to this piece of art that was shown at Uppsala Art Museum. So I am delighted to present Amanda to you, and I hope you will enjoy this episode with her. There is, as always, some pictures related to this on our blog post, which you can find at www.flora-l.com/blog. And with this, I say welcome to Amanda Selinder, and I hope you find this interview inspiring. Welcome to the podcast, Amanda.
Oh, thank you. It's so nice to be here.
It's really lovely to have you here. And you are my first guest who is an artist. So I'm super excited.
Wow.
Getting a new perspective. I've actually realized while reading about you that I didn't really know how artists work with all these things about residency and stipends. And I think you may maybe through my questions tell us a little bit how life as an artist is like as well, in addition to all the other things that we're going to talk about arts and fungi, plants maybe.
Yeah.
So do you want to tell us a little bit about yourself, where you're from and how you discovered your passion for art, and also what your relationship with nature is?
Yeah, I mean, I'm from like a very small town, like 4 hours like south from Uppsala. So it's kind of a small town. So I'm from there. And from the beginning, I mean, when I was younger, I always wanted to be like a musician. So my plan was to become a musician, like up till I was like 21 or something. And then I just wanted to I didn't know what to do. So then I went to like a foundation year. We have like in Sweden that we call like folkhögskola, just to figure out what I want to do. And then I studied art, and it's from there that I continue to work with art and studying at the university and so on. So yeah, so that's how I started. It's not a lot of artists is I started with art when I was really young. It's not like that for me. So it was kind of late in a way.
Mm-hmm.
Yeah.
And your passion for nature, because today you're fusing nature and art. Has that been emerging already as a child, or is that something that you have discovered later?
No, it's also something that I discovered later, because when I did this foundation year, it was more art in general, like more like painting and like sketching and that kind of thing. And then I found like fiber arts like quite early. So I found that fiber as like a material was something that like I really wanted to work with. So then I went to like another foundation year school that was more fiber arts. And there I started to like, I found it quite hard to just work with like a fiber that I didn't know where it came from, or like that it was dyed with something that I didn't know anything about, like a synthetic dye. So that's how I like got into, how is like cotton like made of, like how is it made, like, and then I came into like starting to look at the more like the fibers, how they are produced, and like natural dyes and all that kind of thing. So it basically came from that I questioning where the materials came from and how it's been dyed. So, and then it's like been developed like more and more and more from that.
Such an interesting way. I think also when I look what you are doing today, it's really like you find something and you dig deeper and want to understand more. It's a little bit like doing science. You find something you wonder about and you do experiments to understand more, and you find new questions to understand more.
Yeah, definitely. Yeah.
So when you dye, you are using natural pigments from fungi, also from plants.
Mm-hmm.
Sometimes.
Yeah.
And how did you learn about this? Now we have understood how you discovered this with this idea of how is this actually made, but how did you learn this?
Well, I actually just, there is like, a lot of, literature and books and like a lot of knowledge about natural dyes. So when I started to like make research, I found that, oh my God, this is too advanced for me. So I just started to experimenting in the beginning. So I took like things that I had like around me more, like, like onion skins and stuff like that is like really easy to find and easy to get pigment out from and so on. So that's how I started. And then also, if I'm again talking about when I did like my BFA, but like they had this really, really old cabinet in there, like the first year you do like a lot of dyeing classes and like weaving classes and so on. And then we had this dyeing class, and then I found this big cabinet with like really, really old natural pigments that like nobody had used since the 70s. So I was like, wow, what is this? And that's also how I had like access to a lot of this kind of, rare pigments also. So I just experimented. I didn't really know what it was, and just like, played around a lot with it. So yeah, that's how I like started to work with it.
When I have a book about natural dyeing, and I've read this, but it requires quite some infrastructure to do it, especially when you need salts, usually alone.
Yeah,
salt.
you can use. For me, because I'm more like into the fine arts. For me, it's never been like important that it should like stay for a long time. It should be able to wash like the fabric or all these kind of things. And then I kind of skipped a lot of this like mordanting processes because I just wanted to experiment. Because if you do all this, first you have to mordant the fabric to open up the fibers, and then you need to like dyeing it, and it should be like this specific temperature, and then maybe you have to like mordant it after so the pigment sticks. It's a really long process. So I just skipped like all the steps that I didn't thought was so fun actually. So I just dyed like with different kind of ways to see what kind of pigments that I got out, and then I've been like doing more and more of, depending on what kind of artwork I'm doing, if it should, the dye should stay for a longer time and so on. So
how predictable is the color if you use natural materials for dyeing?
It really depends on what you're using. If it's more like this, I've been working a lot with indigo and woad, for example, and then it gets blue. So that's what you get. But when I've been working with like fungi, I mean, the process is so much longer because I mean, I'm isolating like all the fungi myself. And then, I mean, even if I use one fungi, like I'm stealing from the fungi, it can turn out like different kind of pigments, but it's not what you see. So it's like, yeah, then it gets like a little bit more, not what I expect at all, compared to more like traditional natural dyes. So yeah, it's really different.
It's really like a living organism. You never know what's going to happen.
Yeah, exactly. And that's also why I really enjoy to work with like natural dyes, because I mean, even if you, for example, have like red cabbage, you say, and it depends on like what kind of pH level it was in the soil, what kind of material, I mean, what kind of pH level it is in the water, and so on. So it's so many things that is like affecting the dyes. You can never get exactly the same when you redo it. And that's something I think is really fascinating, because then you have to be more like open what's going to happen when during the process. And I really enjoy and like working in that kind of way. But it's also, I mean, I think it's really nice, but it can also be like hard if I'm going to sell something, for example, because people want to know what they are buying. So that's also hard in that kind of aspect.
Exactly. I was the red cabbage. I'm remembering because our kids, they like when we cook red cabbage to use the water to experiment, to have soap in it and it gets blue, and to have lemon juice in it and gets bright red. So we use that even for cooking our pasta in different colors.
Oh wow. Oh, that's nice. I mean, I've been working a lot with like red cabbage, because I love that you can get like so much different kind of colors just from the same. So I mean, it's great.
It's really fun.
Yeah, that's really, really fun. And it goes so fast. So yeah, it's a lot of fun.
You have spent during your education and also afterwards quite some time in different countries. There was you went to Brazil, you went to Spain, and to New York in the US. What kind of influences has this brought to you when it comes to like natural dyes and doing arts with them?
Yeah, I mean, I did like 1 year like exchange year in New York, and that was actually, I mean, that meant like everything to me, because it was the reason why I wanted to be at this school in New York is because they have, it's more common that you work with like something that is called BioArt. So you're mixing biology and art. And in this school, they have a lab where like a biologist, like educated biologist was working, where you could do your artistic projects. And this was really like an eye-opening for me, like how I, as an artist, can use like this kind of lab methods to make art. So for me, that was like, it changed everything for me to be there. So, and it's also more like they have so many like community labs, you know, that you can work, and it's kind of easy to access to work in that kind of way there. So, and I mean, we don't really, now it's starting to popping up a lot here, but like 6 years ago, it wasn't that common. So for me, that was really nice to be there. I loved it, actually. So that was really nice and helped me a lot.
I feel I need a sabbatical and go there myself and experience that. That sounds really cool. Is there a lot of artists that are taking advantage of this opportunity to work into the biology, and what kind of projects are people doing? Can you give some examples?
Yeah, I mean, it's a lot of, for example, working with like fluorescence bacteria, for example. Some people did that more like, you know, Petri dish art. But then it was like one artist who tried to genetically modify like a tobacco plant, but it should smell like an orange. So it was like a mix of really different kind of project. And there I also started to work a lot with like biofilm, because I was very interested how like producing my own textiles and so on. So I worked for a couple of years with a biofilm, and that's basically like it's cellulose, but it's produced from bacteria and fungi. In that case, that I worked with biofilm, so I did a lot of things with that there.
That's exciting. So, how did you do that? How did you grow biofilms on a large surface?
Yeah, exactly. So I mean, it's basically I used kombucha,
Mm-hmm.
and like the bacterial culture you see on the surface. So I used that one, but I grew like in a really large scale instead. And then I was more like interested in if I was dyeing this biofilm with natural pigments, then you can see, because different kind of like bacterial colonies absorb different amount of pigments. So then it created a lot of patterns inside this like biofilm. So I worked both with the natural dyes and growing the biofilm. And then I was also more in like a very experimental, because I wanted to like see if I could use like my own body as like the nutrient for this biofilm, instead of like, adding a lot of sugar and all these kind of things. So I was like walking around in it and inside like this the bath and so on. And that was like really interesting, because it actually grew like so much faster. And I mean, I'm not a scientist, but I mean, I guess it was because there was so many other stuff like helping this to grow. So that was like more to like immerse myself into like what I was doing, trying to figure out something and so on. So that was quite interesting.
I love that. It's so creative, and there's like where science and art are meeting is a great place to create new things. And how do you get your ideas?
Well, they are just like popping up, and I guess it's more because I started with like natural dyes, and then I get like more smaller and smaller and smaller, like, so it feels like it just pops up. It's always, I know like what kind of direction I want to go, because it's always so much to do. So there's like, when I start with one kind of process, I find something else in the kind of process, and then I start with that, and then I find something else, and then it's just continuing. So it feels like, yeah, I mean, now I'm like mostly working with fungi, but I mean, so it gets like smaller and smaller in a way.
Mm-hmm. Let's maybe talk a bit about the exposition that I have actually seen, where you have been working with fungi and you have been dyeing silk with the fungi. Mm-hmm. Where did the choice of material come from? Why did you use silk?
Well, I mean, I use silk mostly because it's very easy to use to work with natural dyes and silk. It absorbs the pigment very well. And then I also like the transparency. I really like to work with when you're placing like the dyed like fabrics in a room, for example, you get a totally other feeling when you have like silk than if you use like cotton or wool or whatever other kind of material, because it's shiny, but it's also light. So then when you go like between the fabrics, you get like a movement that I like. So yeah, mostly it's because it's the easiest fabric to work with, like in a natural with natural dyes. And then also I like how it looks, and the transparency and the shininess and so on, and how it moves in the room, and yeah.
And it's really beautiful because it has itself a little bit of a like eggshell or creamy color, and the pigments from the fungus were a little bit brownish, if I remember, brownish and towards orange, and it was a very nice combination of different shades on the silk that you had created. And you had there were structures on the silk, so it wasn't just like blobs of pigment somewhere, it was like symmetrical shapes. Do you paint them, or do you fold the fabric and dye the fabric in a specific way, or how do you do that?
Well, yeah. So this, like the exhibition that you saw, I've been working with like endophytic fungi. So I've been like taking small like pieces of different kind of leaves from different kind of trees, and then I've been like putting them on agar medium, nutrition media, like in petri dishes, and then I've been seeing what's coming out, like what is growing out from the leaf, and the one that looks like they have pigment, I've been like isolating, and then like growing further, and so the ones that are producing pigment, I mean, either they can produce pigment like in the media, or they can like the fungi itself have like the pigment, or how you say, and then I've been doing like in different kind of ways. So in one kind of way, I've been putting like the fabric in the nutrition agar media, like in the petri dish, and then I've been putting the isolated like fungi that is like producing or like have like this pigment on the surface. And then it's been growing, and it depends. I mean, the fungi have like different stages. So some, I mean, in the beginning it can be like maybe yellow, let's say, like Fusarium, one fungi in that kind of strain, is more like yellow in the beginning, and then more red when it's like older. So then it creates different kind of colors because, I mean, it's growing on the surface, and then it creates these patterns. But then I also been, when I'm working with like bigger fabrics, I've been like folding the fabric, like you could say like in a shibori in a way, like you're folding in that kind of pattern you want. And then I've been putting it in a big plastic bag instead, and also using like liquid, like liquid nutrition media instead. And then I've been putting in like small plugs of the fungi. And I mean, it's the same thing there, like depending on like if it's in the beginning of its life cycle or like in the end, or if it's like growing inside a pocket of like air, then it creates, so it gets like different kind of colors. I don't really know why, because it depends on the fungi, but I guess it's because how much air it has and how much like like in the plastic bag.
It could be an oxidation process as well that is happening when you have the different pigments, that it's going to be oxidized when there's more air or not when there's less air, and that makes the different color. Like with iron, for example, when you oxidize it gets more of a red color. I think. I don't know which is more, than like the red or the yellow is more the oxidized or the non-oxidized, but it's probably a similar process also with the pigments going on in the fungus. It's very interesting that you have selected endophytic fungi and that you needed to first go through the process of isolating them. And how did you get to that idea? It's not straightforward. I think the most straightforward process would have been to pick fruiting bodies in the forest, which are visible. But so you started with something where you didn't know whether and what they would be growing.
Yeah. I mean, this was actually because, so I did like it's like a public art commission in a specific park in Uppsala. So then it was like the company like Akademiska Hus, which is like, they are like owning the university buildings like in Uppsala and in everywhere in Sweden. And they, in this park, they have been working with like the biodiversity. So they have been like, building like insects, so how do you say, like insects hotel and stuff like that. And this park is very specific because like back in the days, it was part of the castle garden. So it's like very different kind of trees there. So it's a very specific, like unique park in a way. And they've been working with the biodiversity in different kind of ways. And one part of this was to invite an artist to work with the biodiversity in the park. So then I got asked to do this. And because it was like Akademiska Hus, I mean, and they are renting it out to Uppsala University, it was also some scientists there, like researchers that was interested to, depending on what I should do, like be involved in this kind of project. So, and usually in my art practice, I work with things, I mean, that is often around us, but we cannot see them because it's so small. And then I'm growing it in a large scale format. So, because then it's more easier for us to understand that it's actually there. And one thing then was that I wanted to work with fungi, because I mean, fungi is everywhere around us, but we don't see them often. And they are like hidden, like under the ground, like inside cells in the trees and so on. So I knew that I wanted to work with fungi and present the diversity of fungi in this park, even if we can't see them. And then I talked with the Anna Rosling that I've been working with. She's a researcher at the university. And endophytic fungi, because they're exposed to like UV light, they can produce, I mean, it's not always like that, but often they can produce more pigment because they are exposed to the UV light, and it can be a part of the like how they defend themselves, for example, or it can, for example. So that's why I choose to work with endophytic fungi that is like, exposed to UV light. So that's how I started to work with this. And then it was just like a process of like trying to figure out, I mean, I've been cultivating like so much. I mean, it's not like I do like one, sample and then you get like a fungi. So I did like hundreds different kind of samples. And then, I just choose the one that looked like they have pigments or like producing pigments. And I mean, even if some fungi have pigments, they don't necessarily like bind to the fabric. So that's also a process to see if it works to dye with it. So,
it's quite an effort. And in the exposition, you had actually the Petri dishes with the isolates on the wall, so one could see the impressive number. Were they all unique, or were they some that were replicates of each other?
Some were replicates. But it was like a mix. I mean, now I've also had some problems with like, mice and stuff. So then a lot of, and I mean, I'm also learning a lot, like how I should like, how can I keep them, without they get contaminated and all this kind of stuff. And so it's been taking quite some time to do this. And also, I mean, before I worked with Anna, I've also been working with fungi in this kind of way, but more like parasitic fungi, for example. So that is like producing pigments. I've been working with like Fusarium that have like this really bright, like pinkish color, for example. So but it's like, I mean, it's so much more to do with this kind of project. So for me, it's like, wow, it's in the beginning, like really in the beginning. I have so much to do here.
And you have been, when you isolated the fungi, have you been in the lab at the university, or do you do that in your studio?
Well, I mean, before I did like this specific project with Anna, I was in my studio and did all the. But I mean, it's quite hard because you don't have like the flow hood and all these kind of things. So a lot of things get contaminated, and then you're wasting a lot of time. So now I've been working in Anna's lab, and it's also, I mean, right now I'm actually instead of like doing a master in fine arts, I decided to try if I could do like a bachelor in biology. So I'm also studying right now biology, like at the same time as I'm doing my arts. But that is also helping me because then it's easier for me to access the lab. So that's also why I have been able to work in the lab, because I did like this public art project, and then I'm also a student. So then it's easier because I'm still gonna be there, even if the project is, over. Mm-hmm. So then I've been working in the lab with this latest project. And I mean, it helps a lot. It's so much easier, everything.
And I've read you have been continuing with these fungi now to start sequencing them and identifying really which ones they are. What is the aim of that project?
Well, I mean, in one kind of, from the beginning, it was more because it's fun. It's fun to understand the process. But then, I mean, it's also interesting to see what kind of species that I've been working with. So both like in an artistic, but also it's interesting to see this kind of endophytic fungi. I mean, have they been like observed before, like in from another perspective, if they have been, found like in Sweden before, or have they not? And then it's also easier to maybe find, articles about this specific fungi. Maybe, I can get some clues there, how I can work with it. And, so it helps in a lot of different ways. And then it's also, so much fun to do this kind of process. I've never done that before, but now I did like all the steps myself, and now I know the process and so on. So it's I also realized that I can actually do this, by myself, even if I don't have a lab, because there is some community labs that you can use and so on, and easier tools to, and then you can send it in and get like the sequence back and so on. So that's also a good thing, that you can actually do it without having like this fancy lab at the university. So that's good.
Mm-hmm. That's really great. You're very inspiring, I think, how you connect arts and science, and you want to learn more about the science, which is also very intriguing. What kind of doors has this opened to you to know more about the biology of what you're doing?
Well, everything in my art, all my inspiration is like 100% biology, to be honest. I'm an artist, but the people that I get inspiration from, also when I look back, I didn't realize it before, but it's always been like scientists, or like, watching like this nature program about something or whatever. In a way, this like the BioArt is, it's really a new like topic in the arts. So there is like so many things there to like discover. So, and it's also been like, I don't really know how to like explain it, but it feels like I'm not working like in the typical like artist area in that kind of way. I'm like working somewhere else, but I really like it because I mostly like hang out with people that are like into, fungi or like plants or whatever. And that makes me like getting like other kind of perspectives in a way. So I guess that's why I don't really know, actually. I don't think I have an answer on that question.
I think I can relate to that. For me, it's a bit the same the other way around. By looking at arts and by seeing what people do, like to express something, I'm getting ideas for my science. Because it's, as you say, for me, the fact of doing microscopy design or put on fabric, I have also that passion for textile, and to put the things that usually we don't see visibly on a fabric, it's the same to make it visible, and to make it visible in a new way. And I think that is what arts and science can learn from each other. The science brings the depths, but the arts are a way to communicate it in a very creative way. And I think that's just it's so fascinating. And honestly, I wasn't aware that BioArt is a big thing that is growing. So I feel this is really great.
Yeah. So it's a lot of things going on there. So so it's really, really interesting and inspiring, actually. So that's also like something, if I can like, dream big, but because there is no like lab in the art schools today in Sweden. So one of my like dreams is to like create some kind of lab for artists. It doesn't necessarily have to be at the university, but it would be great, because then more people know that it's actually possible to work in this kind of way. So I think that's like in the future. it should definitely be a lab at the art schools. And I mean, if you look at Finland, for example, they have several like BioArt things going on there, like in the university, for example. So it really depends on what kind of country you're looking at. So there's a lot of possibilities.
Yeah, great. Yeah, that things are not existing always gives us lots of possibilities to do something about it. I like that as well. I wanted to know what is next for you. Now you're studying, you're still working with your fungi. What is your next art project that you are doing?
The next like exhibition that I will be a part of is it's gonna be like a exhibition about natural dyes actually at Sörmlands Museum. So it's like the first exhibition where I've been a part of, like where it's actually about natural dyes. So that's gonna be interesting. So I'm gonna be a part of a group exhibition there, and then now it's more that I. How do you say, like leading some projects. So there will also be an exhibition like this year in the end of the year that I will like lead this project, for example, but I'm not going to be like exhibiting myself, but I'm like doing like that kind of thing. So now it's like a mix of this exhibition in Sörmland, and then also to create this new project, and then also do like my studies as well. So that's like what's right now. I've actually been saying like no to some things now, because otherwise I don't have time to do like the studies. I mean, it's so much to do there, and so I have time to finish it sometime.
I'm especially happy then that you said yes to be part of the podcast. What a chance for me. This has been really, really exciting. I think we are coming to the end. I wanted to know where people can find you.
Well, I have an Instagram. That's the best, like where I upload mostly recent things. So I have an Instagram, and it's Amanda Selinder. Yeah, my name, just in one line. Mm-hmm. So there you can find like all my works and so on. And then I also have, if you're interested to see, like when I did this project in the park, I had a specific website that is called biologicalpigments.com, and there you can see like all the processes. So when you get into this website, you will see like a map of this park, and then there are like specific dots, and if you like push on them, and then you see like the process with the different kind of things that I've been doing in the park. I mean, now I talked mostly about the fungi, but I also did some more like traditional like eco printing, like when you're using more like the patterns of the leaves and then like dyeing with them in different kind of ways. And so there you can, if you're interested in like trying to like start to working with natural pigments in some kind of way, you can go in there, and then I have like tutorials and stuff there. So
fantastic.
Yeah.
Is there anything else that I haven't asked and that you would like to share?
Well, I mean, it's been very nice to talk to you, and I mean, fungi is so inspiring. That's why I mostly end up like only talking about fungi when I'm talking about my artistic like research and so on. But that's like where I'm putting like all my energy in right now. It's like so interesting. I really like to work with fungi, and it's so many levels there. So
really fascinating.
Yeah, definitely, in different kind of ways. So
thank you so much for opening our eyes to a new way of seeing and using fungi and working with them, and a glimpse into the world of the fine arts. Thank you, Amanda.
Thank you.
I hope you have enjoyed this episode, and if you did so, please share it. And if you're listening from a platform where you can leave a review or ranking, I would be delighted to get a 5-star review from you. This will help the podcast to be found and make even more people aware of how wonderful nature around us is. If you would like to be updated on future episodes, subscribe to us on YouTube, Apple, Spotify, or Google Podcast, or whatever your favorite podcast provider is. Or register for our newsletter at www.flora-l.com. Thank you and goodbye.